Type 2 secretory pathway is a two-step process. So if I compare it with the type 1 system, in type 1 system, protein can get from cytoplasm to the outside environment in a single step, and it doesn't come in contact with the periplasmic space. But when I am talking about type 2 system, in type 2 system, protein does come in contact with periplasmic space. But somehow, this system is more complex than type 1 system. So, this is despite the fact that type 2 system only traffics protein across the outer membrane. So, जो साइटोप्लाज्म से लगी हुई लेयर नहीं है, जो आउटर मेम्ब्रेन है, जो पेरिप्लाज्मिक स्पेस और आउटर एनवायरनमेंट के दरमियान में है, this system only transfer proteins from that area. And for transferring protein from cytoplasm to the periplasmic space, it uses one of the more generalized system like SAC pathway or TAT system. हमारे प्रोड्यूसर अपनी बेगम के बारे में अक्सर कहते होते हैं कि कम भी थोड़ा और रौला भी ज्यादा. So basically that's what this secretory system is about. So it does very little, but the pathway and the components are way more complex. This is one image in which you can see that SAC or TAT complex on the right hand side is basically used, this yellow, this particular system. Basically this in one go, it transfers the protein inside. And in the second step, I'm writing a big 2 here. In the second step, this complex comes into play and it transfers the protein out. Now, how does this particular whole system goes about? We'll look into it in more detail. So, the type 2 system basically shows evolutionary relationship with type 4 pili assembly. In biochemistry and in biotechnology, we do this very often, that we try to see that proteins from where they have evolved and what are the functional relationships. Sometimes basically it helps us to decipher inside mechanisms in more detail, and we end up understanding them in a better way. If I compare them with type 4 pili, which is present in bacteria, we can see that both machineries share a set of common core components. So they both have outer membrane secretin. They both have cytoplasmic ATPases, because obviously every system have to rely on energy. They have an inner transmembrane protein. They have the major and the minor pseudopilins, and they have a pseudopilin peptidase, also known as methyltransferase. And type 2 systems basically utilize 12 to 16 proteins in order to affect secretion through the outer membrane. Perhaps surprisingly, though, only a couple of these are actually located in the outer membrane. So basically, ये system जो pores use कर रहा है, जो proteins use कर रहा है, actually outer membrane में भी उनकी localization कोई बहुत ज़्यादा नहीं है. और basically जो internal है उससे वो फायदा लेता है. कैसे फायदा लेता है? Let's see. So the remainder of the proteins are located in the cytoplasmic membrane or periplasmic space. So there is this cytoplasmic protein GSPE, which interacts with the ATP and also acts with ATPase, providing the energy to drive export of the proteins. The inner membrane proteins include several, including GSPL and protein M, which anchor the ATPase to the apparatus, and GSPF, which may function to provide a pore for the translocation of pseudopilin into the periplasmic space. The pseudopilin themselves are a group of proteins. The pseudopilin proteins themselves are a group of proteins GSPG2K, which come together to form a large multimeric structure called the pseudopilus. I'm just going to go back to the image if you don't mind. So you are watching two strands. If I can draw on it, you can actually see them very clearly. So one is this strand and the other is this one. So basically these vertical strands are pili. And how do they operate is that protein comes from this side, and ultimately when it comes in front of this particular hole, it pretty much they act as a snooker rod, and basically they hit it outside, they hit it hard, and basically throw that protein outside the cell. That's how this particular protein works. Pretty much now we have an example, let's say a new biomimetic example, where we can see that how proteins play snooker inside your type 2 secretory pathway. So, based on evidence from several experiments, it has been hypothesized that the pilus may grow in order to push, as I was saying that it looks like a snooker cue, push secreted molecules through the outer membrane complex. Or when it wants to close it, it pretty much acts as a cork. Basically, आप लोगों ने जिस तरह बोतलों को कॉर्क से बंद किया होता है, when this particular system doesn't want a protein to come inside or to go off, वो पहले से ही उस होल के ऊपर इस तरीके से पड़ी होती है और वो बेसिकली प्रोटीन को वहां पर जाने नहीं देती. To close off the outer membrane channel when not required. You can see type 2 secretory pathway in more detail, and these are the various protein multimeric units which come together to ultimately assemble this pathway. What these multimeric units are, probably people who are teaching you the course of molecular biology, you would definitely see that they would go into more detail of these systems. But when it comes to biotechnology, there is something what we want to know. So if you are designing a protein which has to pass through this particular complex, you need to know two things: how many copies it has. And we know that the pore forming protein, it basically is a multimeric protein, and there are around 12 to 14 copies. And it is coupled with a lipoprotein, which basically serves to aid the localization of the GSPD multimer into the outer membrane. तो जो लिपोप्रोटीन है वो बेसिकली इसको एक खास जगह तक रखती है. लेकिन आपको ये सारी कहानी सुनाई मैंने, फायदा क्या? फायदा ये कि बेसिकली व्हेन यू लुक एट द प्रोटीन साइज जो आप एक्सप्रेस कर रहे हैं, you have to give the right signal for the right secretory pathway. Because in type 2 secretory pathway, we know that the pore formed by GSPD is approximately 95 angstroms in diameter. So, wo basically, it's a huge pore. 95 angstrom means 9.5 nanometer. तो आपके पास अराउंड 10 नैनोमीटर का पोर है. अगर पोर साइज इससे स्मॉल होगा तो बेसिकली आपकी प्रोटीन नहीं जा सकेगी. सो यू हैव टू सेलेक्ट ऑन द बेसिस. सो आप लोगों का एक असाइनमेंट है, और वो असाइनमेंट इस टॉपिक से रिलेटेड ये है कि गो थ्रू ऑल द सीक्रेटरी पाथवेज एंड मेक अ टेबल इन विच यू वुड टेल मी कि बेसिकली डिफरेंट सीक्रेटरी सिस्टम्स में जो पोर साइजेस हैं वो एग्जैक्टली क्या हैं.